Good afternoon, dear ladies and gentlemen. Welcome to Pharmacogenomics session and in this wonderful personalized medicine networking event. We are now into the second part or the second day of this wonderful event and hope you really made very good contacts and enjoyed the lunch and coffee breaks and the dinner yesterday. And today we meet here to listen to 4 distinguished speakers in the pharmacogenomics area. And we have speakers that are pharmacist background, medical doctor background, clinical pharmacogenomics background. So it's going to be a very interesting topic and very interesting discussion. So please prepare questions because we will have a question-answer session after the presentations. And also, I would like to say that on behalf of organizers to inform you that after this session, you are welcome to Cleaver coffee or lunch area for closing of this conference. So after the session, please go to the Cleaver Sala restaurant for the closing of the conference. But now I think we are ready to start, are we? I think the speakers are very eager to tell you their work and research. And I would like to welcome our first speaker, Aris Saoulidis, senior pharmacist from East Genomics, UK. And Aris is going to tell us about integrating pharmacogenomics testing in routine national health care. And I think it's going to be a really interesting speech. So please welcome Aris.
Hello, everyone, and thank you very much for inviting me here for the honor to speak and share our findings in the UK and the east of England. And today, I really want to talk about how we practically implemented a pharmacogenomic test in the UK, specifically around in the Cambridge area in the east of England. You will see I can't point there, but it's basically the blue area in England on the map you can see there. And this was very much a very big team effort concerted by multiple individuals across the country and across the different regions. And as Professor Una kindly introduced, I'm a pharmacist working in the East Genomic Medicine Service Alliance, which is based in Cambridge. So the outline of my talk is very much around why we had to implement pharmacogenomic DPYD testing in routine care, how we did that, and now what we're doing after it's now been achieved. And when we implement pharmacogenomics, it's very much a concerted effort that has to consider multiple different factors that have to fall into place at the same time almost. And that has to consider how you introduce this intervention into your patient pathways, how you involve patients into this new service, how you fund it and commission it so that you give the best value for money for your patients, how you integrate it into your wider electronic systems and different pathways in a format that's not adding extra work, but it's actually optimizing the work. And it's also very importantly about how you train your staff and your clinical professionals to do this routinely for every patient as part of their routine care. And just to talk a little bit about why we need to do DPYD testing. As a quick reminder, DPYD is an enzyme that metabolizes most of the drugs circulating in the body after administration for fluoropyrimidines. So that's capecitabine and 5-fluorouracil, and less commonly used tegafur. And because DPYD metabolizes 80% of those drugs, it's very important that when you have a deficiency or a problem with this enzyme, this can mean that it accumulates in the body. So fluoropyrimidines have a very narrow therapeutic window, which means it's very easy for them to accumulate and cause toxic effects. So about up to 40% of patients can have toxic events and side effects, and up to 1% may actually have experienced death from this. And fluoropyrimidines then in DPYD deficiency can accumulate and cause this. So it's very important that we have a way to predict which patients will suffer these toxic reactions. And in the UK population and the European population, up to about 9% of patients may have a partial DPYD deficiency. That statistic might be different for the Latvian population, so you might need to consider that in your local implementation as well. And up to 0.1% may have a complete deficiency, meaning they will suffer very severe toxic reactions. So when we look at the genetics more closely, there are 4 most evidence-based variants, and up to 31 variants of unknown significance, and up to 24 suspected additional variants that might contribute to this deficiency. But we're currently only focusing on the 4 variants, and the combination of those 2 variants can lead to a range of activity for the enzyme from 0% to 100%, and it's very much a linear intervention. So if your patient has a 75% activity of the enzyme, you will give 75% of the dose in the broader manner of speaking. So because the evidence has been growing over the last few years, the international body called Clinical Pharmacogenomics Implementation Consortium has published guidance that summarizes this evidence, provides more context and background to the test and the drugs used here, and have provided very clear advice on what to do with your medicine's dose and the choice of medicine depending on your patient's genotype. And this is most important to do in the first cycle, on the first dose. So you need to have the genetic result available before you give the first dose. And then you can titrate upwards according to how much the patient can tolerate. And just to give a bit of history of how this contributed to the UK actually implementing DPYD testing at scale so quickly, this was following the CPIC guidance. The European Medicines Agency realized this was a real issue and published an alert saying that patients should be tested for the DPYD genotype before the first dose was given. So that was in 2020. And as a result of that, they also worked together with the manufacturers of the drug, and they updated the product license to say that patients should have a DPYD genotype before treatment. At the same time, in the UK, with pharmacovigilance monitoring and the Medicines and Healthcare Products Regulatory Agency monitoring toxicity outcomes, there was a coroner's report that highlighted there was lots of toxicity episodes observed with fluoropyrimidines, and something needed to be done. And the MHRA published a similar drug safety update, very clearly outlining the risks of DPYD deficiency that can be predicted using genetic screening before the first dose the patient receives. And that was again in 2020. And as a result of that, and through an excellent coordination effort, the NHS England, which is the Commissioner of Health Services in England, published an urgent commissioning statement saying that they will start funding the genetic screening of the 4 most evidence-based variants. And through a concerted effort, within only a few months, with the NHS and the UK started sequencing up to 40,000 patients per year. So this is available online, this policy, if you want to read more about it. So, okay, the healthcare system is now paying for this test. Now what? The test was also included in the National Genomic Test Directory under very specific cancer indications. And of course, the indications allow more flexibility where you don't have such defined cancer types. We also increased the awareness of this test to healthcare professionals. So there was letters sent out to the different hospitals and different providers of those services to tell them this is now available and it's in context of the drug safety alerts and so on. There was also news communicated so that patients could actually learn more about this test and hear that it's something they can expect in the treatment. So that's very important that it's a two-way discussion. And very importantly, now that this test was available, the clinicians also had a protocol that they could follow and guidance. So in a way, based on the CPIC guidance and through local practices as well, the UK Systemic Anti-Cancer Therapies Board published a guidance specifically for DPYD testing in fluoropyrimidine treatment. And it's very important to note here that this guidance specifically outlines the responsibilities that different staff members in the patient pathway should expect to have in a standard hospital providing chemotherapy services. So, for example, the lead nurse in charge of a chemotherapy unit is responsible to highlight and ensure that DPYD testing is a prominent feature in their pathways and it's being done. Similarly, the consultant leading the unit has to be aware of this test and make sure that it's taking place before they start chemotherapy. And specialist doctors and registrars that are in training usually will go and talk to the patients about this test in more detail, provide informed discussions, and explain it to them. And a pharmacist will usually look at the prescription and make sure that they review it clinically and that the DPYD test has taken place and we have a result, and the result has been acknowledged and actioned and reviewed before the first dose is administered, and the dose is optimized according to the genetic test result. If not, they will have to go back and follow up with the consultant. And the nurse is the chemotherapy nurse administering the drug is the final safety check, making sure that all the previous checks have taken place. So it's very much like a Swiss cheese approach in risk management. So what did we actually do in the East as part of a local and national transformation project? So in the East, we have 18 hospitals that provide cancer chemotherapy treatment, and DPYD tests are sent to the Cambridge laboratory, so it's centralized in the region, and Cambridge reports the results outside to the other hospitals. So as part of a DPYD transformation project, we looked at what we actually do with DPYD testing in 2021. So that was about 6 months after DPYD was already implemented. We looked at how the pathway actually looks like and looked for opportunities to improve it and learn and share experiences between the different hospitals and the country, and then create a framework for future pharmacogenomic implementation. My role was also including about standardizing the role of pharmacists in the pathway. And we also wanted to look at how we can monitor the impact of this huge intervention in the future on patient outcomes. So one example of how we monitored what was going on was that we tried to map the different responsibilities of the different staff members in the patient pathway. So you will see, for example, that different professionals have different expectations and responsibilities of what they do around DPYD testing. So, for example, in the bigger hospitals, they're teaching university hospitals, there usually tend to be some specialist nurses or specialist chemotherapy nurses that take more ownership of managing the patients, and they usually can order the test and follow up on the test results. In all the hospitals and the smaller hospitals, the consultant or the specialist training doctor will usually do this. In all the hospitals, we're expecting pharmacists to check that the DPYD test result has been taken and actioned. So any patients with a deficiency should have the best dose optimized for them. And all of the nurses are expected to make sure that all of these previous checks have been done. Then we tried to map the pathway for DPYD in a very detailed approach and tried to break down every single step that could apply to most hospitals and most pathways. And we found the best way to show that was by picturing this across 4 themes. So the patient experience, how does the patient experience this whole service? How does the blood sample and the testing actually take place? What is the clinical prescribing and screening algorithms for fluoropyrimidines and DPYD testing? And then finally, how do you ensure you future-proof your services to have appropriate follow-up data collection and informed practice? And this also contributed to a national piece of work where we tried to created a gold standard pathway for DPYD testing. You can see this one is simpler because we wanted to apply to all the hospitals as well. And it's supposed to be a framework that hospitals can look towards and try to implement in their practice. Here you can see an example genetic test report of a fake patient. And I just want to highlight that it very clearly says to the clinician, your patient has an increased risk of capecitabine toxicity, in bold writing there. And there's also a dosing recommendation at the bottom of the report. However, we're now moving away from having dosing recommendations on there because the evidence might change. So we're now asking the genetic labs to signpost clinicians to the latest guidance that's evidence-based. There's also some more detailed information on the second page of the report around the test and which variants were tested. So we're only looking at 4 variants. In the future, we might be looking at 30 variants and so on. Some statistics and data of what we have found so far. So over the first 2 years of this service, we sequenced about 9,000 patients. And as expected from the literature prevalence, 6.5% of those patients had one variant of some sort or a combination of the variants. And that means we potentially prevented up to 600 adverse drug reactions. And it's very important in this endeavor that we involve patients and ask them, what do they actually need to know about this whole process? Is there something that they specifically care about that we as clinicians and professionals don't think? Or is there a different way we can do something? And we ask them about consenting. What does the conversation need to involve? How do they experience this? Is this something we need to change? And how do they manage the transfer of information across the different care settings? And one example is that we found that they don't actually want to sign a consent form for the test. They just want to have an informed discussion to explain to them why this test is required and how it's going to inform the treatment. We also published a poster from a national piece of work where we combined all of this data and summarized some of the views of different patient groups that have to do with DPYD testing. And this informed patient information material, which is published on the Macmillan Cancer Support website. And some more opportunities for improvement that we identified is that the patient experience can be improved. So talking to patients more, why we're doing this test, how does that benefit your treatment. We need to figure out a way to systematically document toxicity outcomes and efficacy outcomes so that later you can say, we prevented specifically this number of toxicity events. We also identified that we need to have frictionless electronic systems that talk to each other, and the genetic test result is communicated from the lab directly into the prescribing software. Because at the moment, we're emailing PDF reports, which you can imagine it adds a lot of time between the time that the clinician is asking for the result, the test, and they actually see it in front of them. And then in the future, we want to create prescribing decision support algorithms. Another thing that we're working on is increasing awareness and developing the workforce skills in this area. And just a quick touch on some national projects that we're doing now to continue this piece of work. As I said, the test we're processing around 40,000 tests in the whole of England annually, but we don't know how these tests are being actioned. So after you receive the result, is the clinician and the team taking the appropriate actions? And what's the impact of this test? Is it actually improving patient care? So we're looking at ways to link data sets together and actually create this information and those insights. And we're developing a framework around how we can implement, based on what we learned, the next pharmacogenomic test, for example, TPMT, NAT15, and azathioprine in ALL. And this is an example of a UK-wide audit that we did. And I wanted to point out all the names of the people there. They're not limited to that, but you can see it's a very much concerted effort of multiple individuals to make this happen in the UK, in England, sorry. This was an audit where we looked at specifically the data for 1 week to see how many DPYD test results were actually actioned, so how many led to a dose optimization, and how many were actually done before the first dose of treatment. And we're continuing to collect data nationally with a new project called the DPYD Registry Study, which is a prospective data collection to link patient variation and phenotypes to actual decision-making and effectiveness and toxicity data. And the molecular genetics of ADR study, where we aim to identify patients where we have not found a variant, but they need to be genotyped further, analyzed further to see if they have any novel variants that may be unique in their ethnicity or more rare than the general population. And finally, I want to close with the Clinical Pathway Initiative, where it's a process led by National Genomics Education Program that means we're mapping the pathway of a new service in genetics and identifying every different step that requires some sort of training or education for the pharmacist, the clinician, the nurse involved in that pathway. And these are available online if you would like to have a look at them. And this is how the example looks like for DPYD. So for every step of the process, we identifying the training needs and education needs to create a package for our staff to make sure that they know what they're doing with this test, they're confident to apply it in practice. And finally, just to touch on how interoperability works, this is a very high-level diagram where when a clinician prescribes a treatment, they get a pop-up that will say, have you thought about doing the DPYD test? And then this generates an electronic request that goes into the local pathology system, which then propagates this to the genomic laboratory hub in the region. Then the test result will be pushed back to the clinician's prescribing system and a central database that holds all the genetic information for a patient. And then this central database can push any results and forward them to any other hospitals the patient might visit, or the other GP practice they might visit, or the NHS app that is developing the system. And we've been having some trouble with identifying the optimal way to communicate the phenotype because, for example, the electronic SNOMED codes only have 3 levels of metabolizer status. The CPIC guidance that I mentioned has 5 levels of activity, but the UK guidance has 6 levels of DPD activity. So which one do you actually report electronically? And this is what we're discussing right now. And hopefully in the end, this will lead to optimal prescribing decision support. So we now have one test, but there's about 70 evidenced gene-drug pairs on CPIC alone that we would hopefully like to implement in the future. So you can't expect clinicians to know every single one of them. You need to have some support from clinical systems. Thank you very much.
Thank you very much, Aris, for your talk. Really immense amount of work, and very pleased to learn about your success. And now I would like to invite our next speaker today, and that's Elina Sivina. Elina is oncologist, chemotherapist at Stradins University Hospital. And Elina is going to share experience from Latvia Hospital regarding pharmacogenomics testing. So the floor is yours.
Thank you. Good afternoon, everyone. I'm a medical oncologist from Riga, Stradins Clinical University Hospital, and mainly my work is with patients, and I will try to share my presentations with you because in the beginning of this year when I get this invitation, it was supposed that I will tell about our experience with DPD testing, but unfortunately this project didn't start yet. It will start only at the end of this year, and I hope this practical data will follow next year. Okay, so as we know, the gene polymorphism, as well as a list of somatic mutations, can impact the efficacy and toxicity of cancer drugs. One of the commonly used chemotherapy drugs are fluoropyrimidines. These drugs are widely used since the last century for treatment of gastrointestinal cancers. Many people who are treated with these 5-FU compounds experience severe toxicity. 5-FU have two major mechanisms of action. The first one is active metabolite of 5-FU, prevents DNA synthesis, and the second is the mechanism of action is a DNA damage. So there is three enzymes involved in 5-FU metabolism. Thymidylate synthase encoding gene was shown to be polymorphic with regarding to the variant number of tandem repeats or a single nucleotide polymorphism. A single nucleotide polymorphism of the second repeat in a 3R individuals has been described, leading to a 3 allelic locus. So this 3RC allele leads to reduced transcriptional activity and lower thymidylate synthase levels, but the 3R genotype is associated with a shorter overall survival in the patients who receive this 5-FU chemotherapy drug. So these findings are very controversial, and this thymidylate synthase level and polymorphism is not recommended to test in daily practice. The second enzyme involved in 5-FU metabolism is methylene tetrahydrofolate reductase. The most common are two single nucleotide polymorphisms that lead to decreased enzyme activity. A study in a patient with a primary rectal adenocarcinoma showed increased tumor regression for this 677 homozygotes after preoperative 5-FU treatment. But most studies found that both polymorphisms were not predictive of objective response of 5-FU chemotherapy. And also these polymorphisms are not recommended for testing in patients before 5-FU chemotherapy. And finally, the third enzyme involved in 5-FU metabolism is dihydropyrimidine dehydrogenase, DPD. This deficiency is presented in approximately 3% of all cancer patients, but it is responsible for approximately half of patients with unexpected severe 5-FU toxicity. Mainly, it is a hematological toxicity and even death. Over 30 polymorphisms is in the DPD gene has been identified. Many of them are not clinically relevant. However, homozygosity for DPD2A can lead to complete DPD deficiency, but the incidence of this allele is very rare, less than 0.1%. Due to this reported severe toxicity, the use of genetic testing to identify DPD deficiency deficient patients is likely to cost saving to the healthcare industry. But in Latvia, we don't have experience with testing at this moment. So the next chemotherapy agent I would like to mention is irinotecan, commonly used in colorectal and pancreatic cancer treatment. It is converted by liver carboxylesterase to its active metabolite, SN38. This metabolite is then further conjugated by several UDP glucuronidases or UGTs to yield this inactive SN38G, which is mainly excreted while bile and urine. And after biliary excretion, inactive irinotecan form can be converted to active irinotecan form, SN38, by bacterial glucuronidase, which can lead to gastrointestinal toxicity. So the reduced glucuronidation of SN38 significantly increase gastrointestinal toxicity of irinotecan. The principal UGT involved in the conjugation of SN38 is UGT1A1, with up to 25 polymorphisms have been described, of which a polymorphism in a promoter region consisting of 7 instead of 6 TA repeats is the most common. And the higher number of TA repeats is associated with reduced transcriptional activity of UGT1A1, leading to impaired glucuronidation of irinotecan. And this allele is also associated with Gilbert syndrome. The presence of UGT1A1 28 allele in either heterozygote or homozygote form was shown to be a significant predictor for severe toxicity of irinotecan, with up to 7 times increased risk of severe diarrhea and neutropenia. So the next group is platinum compounds, which are commonly used in gastrointestinal, lung, and gynecological cancers therapy. Platinum-containing drugs form intra- and interstrand DNA adducts, resulting in a DNA replication, inhibition, and apoptosis. The major pathway for removal of these adducts is a nucleotide excision repair. And polymorphism have been described for many of these nucleotide excision repair, but only few are proven to be clinically significant. For example, XPD polymorphism is associated with a higher response rate and longer time to progression in gastric cancer patients treated with oxaliplatinum-based chemotherapy. And the second example is polymorphism in ERCC1 gene. This has been also the mechanism by which this substitution affects ERCC1 activity is unknown. So current polymorphism can be associated with higher response rate to oxaliplatinum, but these data are very controversial, and we do not use this testing before platinum therapy. So the question is what to do with all this information in daily practice, and what should we recommend to our patients. The National Comprehensive Cancer Network guidelines state that irinotecan should be used with a caution in patients with Gilbert syndrome or elevated serum bilirubin. There is a commercially available test for UGT1A1 testing, but the guideline for its use in a clinical practice has not been established yet. European Medicines Agency recommends an initial irinotecan dose reduction for patients with UGT1A1 polymorphism. ASCO guidelines does not provide any guidelines on this matter, but ESMO consensus guidelines acknowledge that UGT1A1 polymorphism is a predictive biomarker for irinotecan toxicity. And it states that UGT1A1 phenotyping remains an option and should be carried out in patients with a suspicious Gilbert syndrome. So there is a clinical example, the case report of patient with a metastatic pancreatic adenocarcinoma who underwent the first line of palliative chemotherapy with modified FOLFIRINOX. Modified FOLFIRINOX treatment is a standard treatment in the first line of metastatic pancreatic cancer in Latvia. So in FOLFIRINOX we have not only irinotecan but also oxaliplatinum and 5-FU, and it is expected that after this treatment is high toxicity rates. After the first cycle, this patient was hospitalized due to febrile neutropenia and diarrhea, and spent a few weeks in a hospital. After recovery, patient received the next chemotherapy with reduced irinotecan dose by 50%. And only after this event, UGT1A1 polymorphism with 7 TA repeats was detected in this patient. So this test is available in Latvia also. So there are also DPYD testing recommendation in 2020. The European Medicines Agency recommended 2 methods for pre-treatment DPYD deficiency testing. In clinical practice, phenotyping using endogenous uracil concentration or genotyping for DPD risk variant alleles. The patients with DPD deficiency are at higher hospitalization risk due to severe toxicity. EMA recommendations have supported the widespread implementation of testing in Europe. This year in ESMO Open were published data of DPD testing before EMA recommendations and after in 23 European countries. Following publication of EMA recommendations, 87% of countries reported an increase in the amount of genotype testing. Implementation of novel local guidelines was reported in 27% responders. Key factors for successful implementation of DPD testing was test reimbursement and existence of clear local clinical guidelines. Latvia still remains in a gray zone. Latvia did not respond this current survey, and at this moment, genotyping and phenotyping is not done before 5-FU therapy in hospitals. And there is a case report, presentation regarding this unexpected 5-FU toxicity. A 65-year-old patient with stage 2 colon cancer after curative surgery received one cycle of capecitabine oxaliplatinum, and 1 week after the start of this treatment, he was hospitalized due to severe toxicity, febrile neutropenia, and anemia. The current patient discontinued adjuvant treatment, and DPD genotyping or phenotyping was not performed, but we can only suggest that such a severe toxicity could be associated with DPD deficiency. So in conclusion, I would like to say that this DPD genotyping or uracil level detection is recommended before fluoropyrimidine therapy, and also UGT1A1 genotyping should be carried out in patients with suspicious deficiency, and I hope that this test will be available in Latvia, and also it is important that this test would be reimbursed in Latvia so that all patients could get information about this, and we could start irinotecan or 5-FU therapy safely for patients. Thank you.
Thank you, Elina, for your insight into these clinical cases. Very valuable information. Thank you. And I would like to invite our next speaker, Professor Renate Ranka. And Renate is going to talk today about tuberculosis treatment, where pharmacogenetics play a very important role in pharmacokinetics and drug toxicity and therapy efficacy and safety. So the floor is yours.
Okay, good day. So thank you for this invitation. And I would like to share some of our results. So what about this drug-induced liver injury or, yes, thank you, or DILI? So it's quite common, and it's known that nearly all classes of medications can cause liver disease. According to Food and Drug Administration, the DILI rank dataset, at least 192 drugs are listed as most DILI concerned drugs, so quite many drugs are there. However, good news are that most of these DILI cases are benign and improve after drug withdrawal. However, it's still important to recognize and remove this offensive agent as quickly as possible, just to avoid this progression to chronic liver injury and even liver failure. Currently, there are no definite risk factors for this condition. However, two are mentioned. Usually in literature, it's a pre-existing liver disease and also some genetic susceptibility. It may predispose certain individuals. So why tuberculosis? This disease is preventable and treatable. However, this tuberculosis is one of the still one of the world's top infectious disease killers, and this disease claims more than 1 million lives each year. Patients with drug-susceptible tuberculosis are most commonly treated with 4-drug combination, and it is widely known that tuberculosis patients may also experience serious drug reactions. This anti-TB drug DILI event was reported in different studies from 2 to up to 28% of patients. And some studies showed that it results in quite high morbidity, mortality, and increased treatment costs. And from these studies, it could be estimated that approximately 1% of those patients treated with this 4-drug combination suffer really significant drug-induced liver injury and have very prolonged and severe this transaminase elevation combined with other symptoms. And luckily, yes, these patients usually recover when the drugs are withdrawn. However, when we're talking about tuberculosis treatment, it's quite important to continue treatment. And here also I put this information from Food and Drug Administration website. And all 4 drugs are listed as most DILI concerned drugs, but especially it's important to note that isoniazid has this box warning. So, isoniazid is considered as the most hepatotoxic agent in this first-line drug combination. Here is quite a nice picture of isoniazid metabolism scheme. It's quite complicated, as you can see, and most of these cases appeared to be because of elevated level of hydrazine. It's with this red dot on this scheme is shown. However, technically, it's quite hard to measure hydrazine level in blood samples. So we opted for other metabolites. It's acetyl isoniazid and isonicotine acid. Well, but it's also known that there are some genetic risk factors that could lead to hepatotoxicity, and it includes different polymorphisms of NAT2 in CYP2E1 and also GSDM1 genes. However, when we decided to look on this subject, these data from studies were quite controversial, and it's probably is related to quite high variability of this NAT2 gene. Overall, all individuals can be divided into 3 groups because there are distinct fast, intermediate, and slow metabolizer phenotypes. And it's quite a long time ago was noticed that difference in this plasma area under the curve level up to 5 times. This difference exists between fast and slow acetylators. Now, with the help of genetics, individuals of each genotype can be classified based on this number of rapid acetylator alleles. This NAT2 allele number 4 is reported as a wild type or normal allele. However, there are reported very many alleles with these substitutions, which could result in decreased function of for this enzyme. So I put on this slide all these alleles which are currently known. So number 5, 6, 7, 10, 14, 17, and 19. Also, this gene is known for high ethnic variability, and so before some studies, you really have to know about in which population you are going to do these studies. So because the frequency of these slow NAT2 acetylators varies quite considerably across the world, here you can see that it's quite frequent in African population and less frequent in Asians. In Europe, these data are quite diverse. And so our first task was to look on these our patients in our Latvian population. So what we found, so we genotyped all these patients, and here is our results about this genotype and allele frequencies. So in total, we found 7 different NAT2 alleles, including several these slow acetylator phenotype alleles. The most frequent was actually this allele number 6, which is the slow acetylation allele. And we really can see that in our patients, there are predominance of slow and intermediate acetylator phenotypes compared with rapid acetylators. Second enzyme, what was mentioned also in this metabolic pathway, is GSD1. Its function, main function are the detoxification of these different electrophilic compounds, carcinogens, also some drugs, including isoniazid, environmental toxins, and also some products of oxidative stress. And it worked by conjugating these substances with glutathione. And so deficiency in this enzyme function is caused by homozygous deletion of the entire GSD1 gene. So we also tested all this our tuberculosis patients, and our results showed that this enzyme deficiency could be observed in half of the patients. Regarding CYP2A2 gene, it's quite a long gene, and there are so many SNPs reported, but without definite, some are without definite functional intentions. So we decided to develop this IGS-based tool for the whole length gene analysis. So it's our approach is outlined here. So we used this PCR-based approach, and now we can sequence this entire gene in every patient, just to have enhance all this information about every possible SNP or indel or whatever. And the last tool required, but it's a very important tool. We needed a tool for TB drug monitoring in patient plasma. Because this pharmacogenetic profiling of drug substances and these corresponding metabolites are really the most informative, because sometimes it's not very easy to tell about these daily cases, yes or no. So we really need to look on the real data from blood. And we designed this LCM-CFC method. And we developed it to suit all these therapy scheme, and it included simultaneous quantification of all 4 first-line anti-TB drugs, including their 6 primary metabolites. And as I talked today mostly about isoniazid, so it was isoniazid and 2 metabolites, acetyl isoniazid and it's isonicotinic acid. So, with these tools in hands, we started our national project, and in collaboration with clinicians. And so we decided to look what's going on in real patients. When we tried to find some correlations between parent drug, isoniazid, and metabolites, we saw no clear picture. It's just like a mess, like a whole cloud of dots. And the same picture we obtained also when we tried to stratify these patients based on GSD1 or CYB genes. However, when we stratified these patients based on NAT2 phenotypes, we can see quite clear pictures. So we have nice correlations between parent drug and metabolites. Also, it was statistically significant that plasma concentrations of parent drug and both metabolites really depends on these Ni2 genotypes. For 2 other enzymes, we did not see such nice results. And when we looked on these clinical data, here we can see these ALAT, ASAT, and bilirubin changes in blood plasma during the therapy in comparison with base levels, so in patients before therapy. And you can clearly see that in 2 patients, these levels undergo dramatic changes. Also, based on this DILI criteria, 2 other patients were also included and matched this criteria. So the DILI in total was detected in 4 patients, and it was more than 11% of our patients. Then we looked also on some different disease factor analysis, and we did not see any relation to age, dosage, and some other factors which are not here, but what is mostly important that we see significantly decreased acetyl isoniazid level in patients with DILI, but only of this metabolite. So no parent drug did not show us some statistical results, and also second metabolite, no. However, this acetyl isoniazid did. And when we tried to put also this genetic background to all this, yes, indeed, all these cases occurred in patients with slow NIT2 phenotype. All 4 patients with slow metabolizers. Some papers, some literature suggests that not all slow alleles are so bad, and some are more prone to induce this DILI and some not. When we looked on this NIT2 diplotypes, so we just see that in our cohort, every patient have different this diplotype, so it's not related to some single allele. And some conclusions that yes, in our study we see that isoniazid has a very large pharmacogenetic variability among patients, and a significant number of patients really experienced this liver injury during the therapy. And we were able to demonstrate this relationship between isoniazid pharmacogenetic parameters and genetic polymorphisms. And also we observed a significant association between isoniazid, pharmacogenetics, and the incidence of DILI. So we believe that this piece of work gives a little bit more evidence that more precise pharmacogenetic-based dosing could improve treatment of TB disease. So some acknowledgements, because this work was a result of collaboration between different institutions and many people were involved. And thank you for your attention.
Thank you, Renate, really sharing the latest results. We saw that they're really just from the lab. Yeah, yes, unpublished data, really interesting to discuss later. Thank you. And I would like to invite Professor Mikko Niemi from Finland, from University of Helsinki, to share his talk and data on pharmacogenetics of lipid-lowering therapy. Very welcome.
Thank you very much. I would first like to thank, of course, the organizers for inviting me. It's a great pleasure for me to be here. I have really enjoyed this meeting and the beautiful city of Riga. It's my first time here, and it will certainly not be the last time here. So thank you very much. So I will be talking about pharmacogenetics of lipid-lowering therapy, or mostly actually pharmacogenetics of statins. So statins, they are essential drugs in the treatment of hypercholesterolemia. About 200 to 300 million individuals take statins every day. And that is for actually a very good reason. They are very effective in reducing cardiovascular mortality and morbidity. However, their very good beneficial effects do not come without some problems related to the therapy. We know that the response to statins varies hugely between individuals. There are individuals who show poor efficacy to certain statin medications, especially at the lower doses, and there are individuals who experience adverse reactions. Even up to 10% of patients using statins experience significant adverse reactions. And very importantly, as statins are used to prevent cardiovascular disease, a very important thing is that even the mild adverse reactions are known to decrease the adherence to the medication, so that patients actually do quit taking statins. We know, for example, from my country, when looking at patients actually purchasing statins after prescription, about 30% of patients discontinue within the first year, and usually the medication is intended for a lifelong medication. So this is a really big problem that patients quit the medication. Well, the adverse drug reaction to statins, especially the muscle toxicity, is a clear concentration and dose-dependent adverse reaction. We know that the risk increases with the doses of statins, especially at the very highest doses of statins. We see that the risk of muscle toxicity starts to increase exponentially. There is a risk also with the low doses, but especially with the highest doses. Then again, we know a lot of risk factors for muscle toxicity and especially drug-drug interactions which lead to increased exposure to statins. They are known to increase the risk to statin-induced muscle toxicity. We have currently in the market 5 or 6 different statins, so we have alternatives for patients, and they do differ. The statins do differ between each other, so there is an opportunity to try to personalize therapy and thereby increase the efficacy and safety of this medication. And we know that genetics and many other individual factors are really important determinants of statin response. A bit more about the statin-induced muscle toxicity. So there is a mild but very common form of muscle toxicity which we call as myalgia or muscle pain, which occurs in about 5 to 10% of patients taking statins. It's usually not medically kind of a dangerous adverse reaction, but the danger relates to the poor adherence related to this muscle pain that the patients experience. On the other extreme of this toxicity, we have the very severe forms of muscle toxicity, rhabdomyolysis, which occurs in about 1 per 100,000 patient years of statin use. It's a very rare adverse reaction, but as you know that there are hundreds of millions of patients taking statins, there are these severe cases every year in even smaller countries, like in Finland, we have about 700,000 patients using statins every day. So we see 5 to 10 rhabdomyolysis cases every year due to statins. And then there are even more rare forms of muscle toxicity, which have maybe a slightly different mechanism underlying, but all the other forms, they are clearly dose and concentration dependent adverse reactions. So my interest in statin pharmacogenetics and statin toxicity started actually more than 20 years ago. When I was finishing my PhD thesis on drug-drug interactions of oral antidiabetic drugs, my colleagues were actually investigating drug-drug interactions of statins. And they had noticed huge inter-individual variability in statin exposure, so the concentrations of statins, even in healthy volunteers without any medication. We saw more than 10-fold differences between healthy volunteers in the exposure of statins after the same dose. So there had to be some individual factors other than drug-drug interactions explaining these differences. So what we found, we went on to investigate was a certain transporter which was identified a couple of years earlier by actually 3 groups around the world. They identified a transporter which we know today as organic anion transporting polypeptide 1B1, OATP1B1. It's a liver-specific transporter which pumps its substrates from blood circulation into the liver. And statins were actually one of the first drugs identified as a substrate of this transporter. Of course, the site of action of statins is within the hepatocyte, so it's very good that they are taken actively into the hepatocytes. But we wanted to investigate whether we could identify genetic variability in this transporter that could explain these huge differences in statin exposure that we had identified. And in fact, we found a common single nucleotide variation in this OATP1B1 transporter, which results in practically non-functional transporter if a person is homozygous for this variant. We investigated the effects of this variant on different statins, and we found that indeed it does increase the concentrations of multiple statins. The most marked effect we see on simvastatin, one of the most widely used statins. Its exposure is increased more than threefold in individuals homozygous for this poor function variant. A couple of years later, there was a genome-wide association study about statin-induced muscle toxicity, and the only gene that was genome-wide significantly associated with this muscle toxicity due to simvastatin was the same SLC1B1 gene encoding for this hepatic transporter. And in fact, the functional variant or the causative variant was found to be this exact variant which causes poor function of this transporter. So the evidence was actually very, very good already 15 years ago. We knew exactly an important genetic factor predisposing to statin-induced muscle toxicity. We actually introduced this genetic test, I think, in 2009 in our university hospital, and it has been available since that. However, you can imagine that the world is not that simple, that we have one gene and one adverse reaction. It definitely is not that simple. There are multiple other genes affecting the response to statins. Regarding the efficacy of statins, we know that there are at least 4 important genes related to the cholesterol-lowering efficacy of statins. These are data from a genome-wide meta-analysis by a British group identifying these 4 genes. So 3 of these genes are related to the pharmacodynamics of statins, so the effect of statins on cholesterol. And the 4th gene is this SLC1B1 transporter. So the transporter is not only important for the toxicity, it's also important for the efficacy of statins, which we can understand as this is an influx transporter pumping statins from blood into the liver. So when the transporter does not function, we can understand the efficacy of statins is reduced. However, at the same time as the systemic exposure is increased, the risk of muscle toxicity is increased in association with this variant. So this is all very exciting. We have actually recently continued our work on statins, and what we are trying to build is to try to build a comprehensive understanding of all the genes and how they affect these different statins, as they actually differ. There are differences between the statins, how the genes affect them, especially their concentrations. We have looked at the pharmacogenetics of statin pharmacogenetics, so the concentrations of statins in healthy volunteers in clinical trials of up to more than 200 healthy volunteers. So we have looked at all the statins on the market. We have already published the data for simvastatin, rosuvastatin, and fluvastatin, but we still have data for atorvastatin, pravastatin, and also pitavastatin, which is not very widely used. We have done either a genome-wide association study for the statin pharmacogenetics, or then we have done next-gen sequencing of all the pharmacogenetic genes to try to identify really the most important genes related to statin exposure. Important things are that there are differences between the statins. The OATP transporter, for example, its poor function has a different effect on different statins. Simvastatin is the most susceptible statin, and fluvastatin is the least susceptible statin. When we found out this already actually 10 years ago, we thought that, well, this actually forms a good basis for individualizing statin therapy. But then there are these other genes as well. For example, another drug transporter known as breast cancer resistance protein encoded by the ABCG2 transporter or gene, and which has an impact on one of the more modern statins, rosuvastatin, and here a poor function in the transporter results in increased absorption of statin from the gut, rosuvastatin from the gut, and therefore we see increased efficacy and increased risk of toxicity in association with poor function of this transporter. So we have three genes affecting the concentrations of statins. So far, there are a couple of more coming actually from our unpublished data. And then we have 3 genes affecting the pharmacodynamics of statins. And how do we then actually make these into the clinical practice? How do we get them to be tested, first of all, as we have the DPYD example in the first talk you heard, and how do we get them to be interpreted correctly in the clinical setting? So I've been very much interested in clinical implementation of pharmacogenetics and clinical implementation of statin pharmacogenetics. So we heard in the first talk, there was a very nice overview of what we need when we start to implement pharmacogenetics. So one of the things is guidelines. How do we interpret the results? I have been very happy to work with the Clinical Pharmacogenetics Implementation Consortium with the statin guidelines, and we published last year an updated guideline for statins, where we included the 3 pharmacokinetic genes affecting statin exposure and came up with guidelines for all these different statins, how to interpret the genetic information. We have maximum recommended doses for different genotypes, and we can recommend avoiding a specific statin for individuals with certain genotypes. So we need the guideline, but that's not enough. We need tests available in the clinic. That's the next step. And what we have done in our university hospital, so I work at the Helsinki University Hospital and its diagnostic laboratory, we call it the HUS Diagnostic Center. So this diagnostic center, we serve a population of about 2.3 million, so about the size of Latvia. And we, even though this is a university hospital lab, we serve the entire healthcare from primary care up to specialist healthcare, and we do practically all the laboratory tests for the population of this southern part of Finland, about 2.3 million. The hospital sees about 700,000 patients a year, so it is a really big hospital in our setting. So we have been doing pharmacogenetic testing since 2004. We started with TPMT testing for childhood leukemia, and this has been in routine use in Finland since 2004. After that, we have introduced several additional individual tests for different genes. CYP2D6, we introduced the SLC01B1 test for statins, as I told you, more than 10 years ago and so on. But in the last, let's say, 5 or 6 years, we have seen a huge increase in the requirement of pharmacogenetic testing from clinicians. So the number of tests was increased, and there was really a need to introduce new genes also into our panel or into our test pattern. So we decided, I think already 4 years ago, that there's no idea actually for a clinical laboratory to have these 10, 12, 13, 14, 15 different pharmacogenetic tests. We should come up with a panel test to include everything at the same time. I said we decided, but I have to say that I'm the culprit. So I decided this, and it's not always easy to push things forward, but this was finally successful, that we went on to develop a panel test. So what we did, we wanted to create a as cheap as possible test with a high throughput, low turnaround time, and so on. And we decided to go with a next generation sequencing technology. So we have a targeted NGS panel targeting these 12 genes related to pharmacogenetics. It's highly automated, high throughput, and we have a highly automated system of also reporting the results electronically to the laboratory systems of our hospital area, but also actually we are making the test also for all the other university hospital districts in Finland. So we are covering the entire country with our test. When we were actually trying to rationalize for us whether we should go with a panel test or we should go with individual tests, we did some background work with our hospital patients. First of all, we wanted to investigate how common this variability is. It's very well known that pharmacogenetic variability is very common. Looking at this 12-gene panel and the Finnish population, at least 99% of the population carry at least one actionable genotype. So an actionable genotype means that if you know that a patient has this genotype and you are starting a specific medication, there is a guideline that suggests you should either reduce the dose or select an alternative dose of the drug or then select an alternative medication. And there is good evidence supporting this guideline. So this pharmacogenetic variability really is very common in practically all populations. Also, the medications are quite common. Very briefly, what medications are important in relation to these genes? There are antidepressants, there are anticoagulants, there are the statins. We have the 3 genes for statins. Actually, one thing related to the implementation last year, or actually this year, there was a current care guideline in Finland that recommends testing for pharmacogenetics for antidepressants in certain situations. So this is very important to have national guidelines that direct physicians to start testing in certain situations. There is very good evidence in support of using pharmacogenetics when selecting an antidepressant. And there is very good evidence also suggesting that this is cost-saving in most cases. So this is why we have this current care guidelines. So we have also from psychiatry, we have interest in pharmacogenetics. There are other drugs, there are analgesics, there are the fluoropyrimidines mentioned already, there are thiopurines mentioned already, and so on, so multiple drugs. Coming back to actually statins, there is another very important drug in this slide, clopidogrel, which is an antiplatelet agent. In the case of clopidogrel, we have a very good evidence that poor CYP2C19 function or even reduced CYP2C19 function is associated with poor efficacy to this medication. And there are tens of thousands, if not hundreds of thousands of patients for example, in my country, taking statins to prevent the recurrence of a stroke or a myocardial infarction, and they don't know if their genes are actually compatible with the medication. A couple of years ago, there was a randomized trial showing that actually genotyping patients after myocardial infarction and selecting the antiplatelet agent based on the genotype, it has benefits for the patients. There is a reduced risk of bleeding if you select the medication based on the genotype. So this is what we are currently doing at the Helsinki University Hospital. We are running a pilot study. It's not actually a pilot study, it's a clinical pilot where we are genotyping all patients who come into the hospital with a myocardial infarction. We do the panel for the selection of the antiplatelet agent, either clopidogrel or ticagrelor or prasugrel. And in addition to this, they get the information for statins as well for free. So with the same test, the patients now actually benefit also with regards to statins. And this, I think, is the way to push pharmacogenetics forward. So when you have an indication for testing for certain gene, if possible, try to push for a panel testing so the patient gets actually all the other genes as well in addition, and is likely to benefit from that. I think my time is coming to an end very soon, so I'll try to be very quick with my last slides. But before going to those, I must say that when we looked our university hospital patients at the Helsinki University Hospital and the medications that they were prescribed while they were inpatients in the hospital, but 40% of them were using at least one drug which has an actionable pharmacogenetic guideline. So 40% of our 700,000 patients yearly would potentially benefit from pharmacogenetic testing. Actually, our hospital lab made a press release about our pharmacogenetic panel last year, and they interviewed me for that, and I said in the interview that, okay, about 40% of our patients could potentially benefit from pharmacogenetic testing. And then it took about 2-3 hours, and I got a phone call from the hospital directors or one of the directors of our hospital, and he wanted me to know that, or tell him that how are we actually going to pay for the testing of about 300,000 patients a year. So there is really a question about cost effectiveness and costs, and we have to really be careful when we are starting to do large scale implementation, and we have to be really sure that what we do is beneficial for the patient, but also cost effective or even cost saving. There is a huge potential even for cost saving with pharmacogenetics, so I don't think it will be an issue at the end, but currently we are doing pharmacogenetic testing on certain special occasions, let's say. Coming back to statins, so we know the pharmacogenetics of statins quite well. We know what genes are important and so on, but it's not only the genes, as I told you in the start. So we have many other patient factors that are important for drug response, and that's also true for statins. So there are the drug-drug interactions and many other individual factors. And how we are trying to address this is to build a systems model for statins. So we are trying to incorporate all of our knowledge of the factors that affect the exposure, efficacy, and toxicity of statins and build a model to predict the response to individual statins at the level of an individual patient. This has been a 6-year project and it has come actually to an end, so we are now finalizing our model. It really took us 6 years to come up with the mathematics of this model, but we are now finishing it, and our next step is to bring this model into a clinical trial. So we will be running a trial on a randomized trial to test whether actually selecting statin based on a prediction from the systems pharmacology algorithm actually benefits the patient. So our aim in the trial is to reduce the number of patients who discontinue the medication. So there we have sufficient power to detect an effect. So to summarize, we know certain genetic factors which predispose to statin-induced muscle toxicity and which can affect the cholesterol-lowering efficacy of statins, and especially panel testing in the case of statins, I see is a very good way to start implementing statin pharmacogenetics in the clinic. It may not be cost-effective by itself to test every patient who you are starting with a statin, but when you get the result for free, when you are testing for other reasons, then it may become cost-effective. With this, I'd like to thank you and use 10 seconds to advertise 2 meetings. So next in 2 weeks, we are going to have the conference of the European Society for Pharmacogenetics and Personalized Therapy in Copenhagen. I hope to see some of you there. I know that at least some I will be seeing there. And in 2025, we will be hosting the European Association for Clinical Pharmacology and Therapeutics conference in Helsinki. It's a short way from Latvia, and I really hope to see many of you there. With this, I'd like to thank you very much for your attention. Thank you.
Thank you, Mikko. Very inspiring. And now I would like to invite all speakers to take a seat in a panel, and we will start a panel discussion. And of course, the audience is welcome to ask questions to our speakers. So, and there will be some help with microphones, I suppose. Yes, first question is there. Welcome. I thought you will be the one asking question. Well, actually, I have light in my eyes, so maybe anybody wanting to talk, just lift up hand and the microphone will. Yeah, there is a hand here. I see. Yeah. Okay.
Thank you. As a clinician in the field of medical oncology, I will address my question to Aris, the speaker from first presentation. I just wanted to ask, maybe this advice would aid my colleagues as well. If we don't have opportunity to test these beautiful DPYD polymorphisms, then from your perspective and practice, what would be advice on initial dosage of medication if we don't have results of DPYD testing? Should we go with 100% dose, 75%, or be more cautious? What would be your advice from your practice? Thanks.
Can you hear me? Yes. Yes, thanks. That's a great question. And I think that it largely depends on, of course, the tumor type and, of course, the clinical area that this applies to. But I think the clinical practice that our oncology team are applying is if for example, if there's a case of emergency, if there's an urgent treatment that the patient needs to receive before the result is available, or if for some reason the result is not available on time, they typically tend to start with 50% dose reduction. And I think then they tend to build up the dose in increments, perhaps of 12.5%, 25%, as the patient tolerates with each cycle. And it's very much dependent on the patient and how frail the patient is. As you know, and it has to be a professional judgment for every personal patient and personalized.
Thank you.
Yeah, anyone has a question, please? Yeah, there is a question. Welcome.
I'll try to. I don't know to which side to go to see my Finnish colleague. Actually, about the pharmacogenomics and your studies in Finland, I'd say that I'm a clinical pharmacist, so usually my daily work is connected with not only the antimicrobial resistance, but as well like with the interactions, different like medical therapy. And the problem is that basically it's like the fortune telling of the interaction levels, not only among the medicines, but actually when we take into account our hepatic enzymes, or like the enzymes and the metabolism, it's really a fortune telling in Latvia because we don't have these data like you have and Estonia has. And the thing is, basically, according to the data, I can assume that the Balts and the Finnish people basically are more slow metabolizers rather than rapid. So in terms of therapy, I should assume that we are slow in most of the cases, if I tell the fortune of taking into account the metabolizing features. So what could be your remarks until we get our genome results in Latvia? I don't know in how many years.
Yeah, thanks. I think this is very important. And it's very important to understand that these are really population specific. So the Finnish population, you've heard that we often talk about non-Finnish Europeans and Finnish Europeans. So for example, our Finnish population differs actually quite a lot from other European populations with regards to the frequencies of pharmacogenetic variants. For example, in the case of cytochrome P452D6, which is important for codeine and antipsychotics and antidepressants and so on. So in Finland, we have a very high frequency of an ultra-rapid metabolizer genotype. So the frequency is about 7% of the population are ultra-rapid, whereas in other European populations, usually 5-6% are poor metabolizers. So we are completely the opposite in this regard. So it's really fortune telling if you don't have the data. So I think the data are very important in this regard. Regarding the drug-drug interactions, I can understand your difficulties with that. So there are so many drug-drug interactions that one has to deal with. So databases are very important tool in this regard. For pharmacogenetics, as was mentioned, I think in the first talk, we have about 70 gene-drug pairs, which are important. So that is actually, it's not that much information. So it is currently, I think it is doable without clinical decision support. And in Finland, in my hospital, we have set up a guideline or a booklet of pharmacogenetics for clinicians to look for the information what to do with which medication. But we are also looking forward to building these decision support tools. I think that's the future. But you can do even without those with pharmacogenetics.
Could you share that booklet at least?
Yeah, it's freely available. It's in Finnish. So if you Google, unfortunately it's in Finnish, but you can try and translate it. But it's basically based on the CPIC guidelines, which are also freely available. So the information is available, but the work that has to be done is to make it kind of localized to your local language, but also to your local clinical practice. So that is the work. It took a few months for us to do that, so it's not unthinkable to do that.
Just a small question. Do you have only in Finnish or in Swedish as well?
Currently, we don't have it in Swedish, but that's what we are going to do because we are a nation of two languages. Yeah.
Okay, thank you.
Thank you, because I had one of my questions too. If we could directly replicate the panel from Finland to Latvia, and your answer is that no, it's like based on this local or ethnic or population genetic variants.
Yes and no. So our panel, actually, we designed it so that it's globally comprehensive. So it includes practically all functional variants that we know in any population that have any frequency in some population. So it's very comprehensive. So you might be able to replicate it, but I have to say that it took about 2 years for us to set it up. And the big thing was not really the wet lab, it was the bioinformatics and so on included in there. So there is huge amount of work to set it up.
I see that, and it's a huge amount of work, and then actually lots of different types of people and professions involved, like bioinformatician, pharmacist, clinical doctor, researcher, patient, like a lot of like big turnaround and many people involved, and there has to be like clear path. And I think the path through that you Aris demonstrated is really nice, and that kind of also can serve as a good example how to do things. But I understood that in UK there is currently just DPYD guidelines and use in clinics, or is there any other genetic marker used, pharmacogenetic?
Absolutely, yes. So we have the National Genomic Test Directory, which is split up into the cancer, rare disease, and now that we have the pharmacogenomics theme as well. And it depends on how you define a pharmacogenomic test. So if you're doing a molecular test for a cancer type or a rare disease type, usually most of these will inform your medicine choice and medicine optimization. But we tend to call these precision medicine. But what we call a pure pharmacogenomic test is like a DPYD test, for example. We have 4 of them currently on the test directory. So that's DPYD for fluoropyrimidines, TPMT and NAT-T15 for azathioprine and leukemia, and MTRNR1, which predicts patients who will suffer from aminoglycoside-induced autotoxicity. And we are considering different approaches on how to implement such a comprehensive panel, like the examples that we heard, that cover most of the common medicines in the population. But we really need to think about it very carefully, because the amount of tests alone that is going to be in the millions. And it's about how you implement that at scale, what's the throughput, the affordability, and the cost effectiveness. So once we work all that out, hopefully we can also push it into clinical practice.
Right, absolutely. So any more questions? Yeah, but I like this idea that also goes in line with what Mikko said, that you do some other tests and then along with these tests you include some pharmacogenetics. So it's not just pure pharmacogenetic testing, it's that in line with diagnosis, with the selection of medicines, and also takes some of these tests along. Yeah. Any question from the audience? Yeah, but I'd like to also say that I was very interested in Elina's talk about these clinical cases, because you also gave your recipe how you deal with patients without knowing the genetic background, is that you simply adjust the medicine, the dosage, you look for alternative treatments, right?
Yes, thank you for your question. I would like to say that we usually start with 100% of dose. We do not start with a reduced dosage of 5 fewer irinotecan. And then if the patient experiences some toxicity, then we can reduce the dose. Yes, but usually we start with a full dose. Also, I would like to say that in our clinic, in our daily work, we have so much factors which could impact the toxicity, the outcome, response. The patient factors, is this patient living far from hospital? Is he living alone? There are some relatives who are helping him with these toxicities. Also tumor factors, somatic mutations. There is a list of different factors, and pharmacogenomics is only one little thing in all this list. Of course, if we would have this test reimbursed, we will do this daily, and we could also, as it previously was mentioned, save the money, of course, with this testing. But in real life, we are working with these full doses, yeah.
Okay, thank you. Yeah, and then going next to Renate about tuberculosis therapy and this liver toxicity. How was your collaboration with clinicians started? Did you approach them, or the clinicians approached you with this problem in tuberculosis therapy with toxicity?
Oh, it is quite a long journey before. So it was, coffee breaks, a lot of coffee breaks, and decide what to do. And finally it was. So we succeed to with these fundings. So it was made all these available, and we were clear to go.
Right. Do you see that this test will be approved, like clinically, how do you call it, certified, or? Yeah.
Well, it depends, of course, but our goal was to put some more information on this topic, because the results that are in literature are not so clear, and so we hope with our data it could be became more clear about practice. Maybe yes, sometimes in future, but not. It seems that not clearly only genetics. It should be together with some other biomarkers, for example, drug levels or whatever. It's too diverse, these levels, what we can see.
Yeah, thanks. I agree. It has to be looked more complex. It's just a single parameter of pharmacogenetics. Any comments from the audience? Sharing experiences from clinics? If not, then I should say that I learned a lot from you. I'm a university lecturer, and I teach students pharmacogenomics and pharmacogenetics, and certainly I got a lot of information. I was typing all the way, and that I will tell students about experience from Latvia, experience from UK, from Finland, and also you saw the advertisements of next meetings. So certainly more coffee breaks, more new ideas, new projects that I see. And I'd like to thank speakers again with applause. And also thank you, the audience, for your interest, and also your questions. And there's opportunity to ask more questions during the coffee break before just closing ceremony. And welcome to the restaurant area for conference closing. That's a pity to announce this, but this is what it is. Thank you for these wonderful 2 days together. Thank you.